Yeah, so a little overview. So sodium channels are basically membrane proteins, and proteins are found all over the body. A lot of stuff we have are made of proteins, our enzymes, our hormones, neurotransmitters are inherently proteins at that. The structure of our membranes, structure of a lot of things in the body are as a result of proteins. And then proteins come in the form of nutrients. Maybe proteins are just so great and so vast. Um, but of course, they're membrane proteins you can find in neurons, and specifically when talking about sodium channels, we'll get into a bit of what sodium channels are, what that comprises of, what everything is. I'll briefly be able to dive into that in this presentation for you guys. So they play a crucial role in the generation and propagation of action potential. That means carrying out neural impulses. And we should know the basics probably of what that means. So these channels are highly selective for sodium ions, which are basically sodium atoms that lose an electron, exist in various forms, but one of the most well-known types in neurons is the voltage-gated sodium channels. So voltage-gated sodium channels are the basic ion channels for neuron excitability, because neurons can, of course, excite, expend energy, and, of course, inhibit resting. So, which are crucial for neural impulses or action potentials, I prefer to say, in neurons. So there are at least 9 isoforms detected in the nervous system. You have like NAV1, that go up to NAV9, whatever. But recent studies suggest that these channels play a vital role in the electrical activities in neurons and linked to various neurological diseases such as epilepsy, brain tumors, and neural trauma, and MS. So MS is basically the myelin sheath. This is a layer of fat, I would probably prefer to say, around the axon. The axon is basically the long tunnel that carries your neural impulses to be able to go to the cell body, the dendrites, to another whole neuronal cell. The hippocampus now, I like to focus on, because we're focused on sodium channels in neurons, of the hippocampus in specific. It's a brain structure that's pivotal for memory, spatial navigation, contextual processing, and the integration of emotional experience. So you can see how it's a big loop of like this broad network of all these different brain regions. So you have, since it's the center of memory, memory can relate to emotions. Emotions can be related to the amygdala and the insula. And then you have memory in terms of learning that can be related to a lot of the cerebral cortex. You have even motor skills that can be linked to the cerebellum, linked to the sensory motor region in the parietal lobe. You can see how even the hypothalamus can be connected to, because that's regulation. You can see how all of these different things still tie into that basic memory, and then the navigation part of it, and then the learning part of it, and the emotional part of it, and the stress part of it. You can see how that's all vitally linked, essentially. So introduction to the hippocampus. The hippocampus is a small seahorse-shaped structure deep within the brain, as you can see here on the diagram, and I have the reference of where I got the figure image from on here as well. So, of course, damage to the hippocampus leads to all these different impairments, as we all see, and that I spoke about in the prior slide. And, of course, with dementia, dementia is when, basically the death of neurons, basically necrosis on an advanced level. That's usually seen more in elderly people as you age. Cell regeneration isn't as good as it used to be. Neurogenesis, neuroplasticity isn't as good as it used to be for various reasons. That can be either biologically going on, it can be mutations going on, it could be not like this regulation going on. Then it could be lifestyle things going on. I mean, you're not actually forming these new neural pathways because you're more sedentary. All these, you're not learning new things. All these type of things can be going on. So, of course, the hippocampus, what does it actually do? The primary function of this, this seahorse shape is relatively small compared to the overall size of the brain. But, of course, it's interconnected, like I said, with other regions of the brain. We know the amygdala is responsible for a lot of emotional responses, usually negative emotion in terms of fear, excitability, these different things. And it's also in that basal region, as you can see here in the midbrain. The entorhinal is, and the insular cortex is more associated with like emotional awareness of others and emotional awareness of self. So you can see how all of that ties in with those three structures. So memory, of course, is the formation of new memories, particularly episodic, which means story, declarative with like factual information like that's black, that's white, this is what it does, that is what it doesn't do. Then you have other forms of memory too, that like procedural, some other ones that aren't listed into this. Then you have short-term, long-term memory. And of course, short-term, long-term memory is based on the strengthening of that neural connection, which basically means, on a broader spectrum, that means like the more you study something, learn something, the more you're exposed to something, and the more you tie some level of relatability or prevalence or relatively to something and importance, then of course you deepen your emotional attachment to it. You deepen your imagination about that specific thing, and that forms those strong neural connections on a biological level. Navigation, like creating mental maps in your head. We all know when you go places, you'd be like, well, I know where the store is from here. Or I know where that country is compared to relative to my location. All this is due to the hippocampus, and of course other brain region that it works in connection with pattern separation, being able to separate things, identify things, pick things out. Because if everything was the same, we'd be able to identify things. So that also, hippocampus also aids in that. Contextual processing, meaning like being able to bridge gap, connect dots with things, is obviously related to hippocampus. And you can see how memory is tied into that, because based off prior experiences, based on memories, you can be able to see, I have that with that. And also, it's a great saying that usually can be able to connect dots based on things that already happened more than predicting ahead. And a lot of times, like after something happens, because it happened, you now have that memory about it, and also it's connected to other things. You can be like, okay, now that makes sense. So we have learning and flexibility, amnesia, stress regulation, and various other structures. So what even are neurons? So of course, neurons are basically cells that basically are created from stem cells. You could even say that. There are estimated to be 86 billion neurons. And of course, this number may not be exact, but it's an approximate estimate, like it says. And, a developing baby must create a lot of these neurons. So 250,000 is relatively small, I guess, to how much is needed to that stages of development, and development through from embryonic stage to the fetus stage to the delivery stage. All that is important. And then childhood, teenage, that adolescent stage to a full well-off adult, which doesn't get around until about late 20s. It's vital and pivotal for the overall function of a good nervous system. So we looked a bit about, some research on the hippocampus. This is more of a detailed image about the structure of the hippocampus, as you can see here. So on to talking about ions and ion channels that you can find in neurons. And specifically, we're talking about hippocampal neurons in this. So an ion is basically an atom. And you can have a group of atoms. A group of atoms usually referred to as molecules anyway. But when the atom has a positive or a negative charge attached to that atom, it now becomes an ion. And an ion has two categories. They have cations and anions. Inside the cell of a neuron, it'll be more anions, and that gives it that negative charge. And as you can see, what ions are. They come from atoms. They're charged atoms. And so you understand. And when it comes to body and cells, it's a bunch of atoms and elements, and it's so many subunits and everything just floating in it's basically like a space or an ocean or a soup or just a vortex of different things just floating around. And a lot of stuff you can say is random, but they all have specific instruction and codes and structures that help it be able to bind where it needs to and do exactly what it needs to. But it's all just randomly floating. So that's kind of the origin. They all are currently existing there. But we can talk about the external things or endogenous things that you can say that need to enter the human body. But most of the things we have to essentially survive are here in terms of functioning of cells. But of course, it's the same that thing. We have to get various external things. But ions and everything of that are really highly prevalent in the body. So we know neuronal ion channels are gated porous. They open and close like a door, and they are regulated by voltage. Voltage basically charges that we see positive and negative charges that basically you can relate it to power. It's all things that we all are kind of familiar with in our everyday seeing life. So of course they're permeable to ions such as sodium, potassium, and calcium. And rapid signaling in neurons require fast voltage sensitive to mechanisms for closing and opening. Small changes in gated properties of a channel that can have profound effects. Extremely low frequency electrical and magnetic fields are thought to produce at most microvolt changes in neuronal membrane potential. So we know potential is basically like information being passed, and you can be able to measure that. It's basically, you can say it in probably the most simplest way. So you have different proteins that carry, but these aren't specific proteins that are structural proteins essentially. So we know about all the basic introduction about it up to now, or we should know. But we know that they mediate fast depolarization. So depolarization is basically when the charges go up and it basically becomes more positive and it excites the inside of the cell and it sparks be able to go down that axon terminal to be able to send whatever information it needs to send to activate whatever it needs to activate, whether it's to send off neurotransmitters or activate signaling for hormones or activating proteins, all these type of things. So more about the sodium channels. There are two major classes of sodium channels in mammals, and we are mammals. The voltage-gated sodium channel family and the epithelial sodium channel. So voltage-gated sodium channels exist all throughout the body in various types, while epithelial are most located in the skin. And we know we heard of epithelial tissue before. I would assume if you've done any type of biology, you probably heard the term before. So these, of course, allow the flow of sodium from the region of higher concentration with its extracellular to a lower concentration where it's that would be intracellular, and there's more potassium inside than outside, and it's more sodium initially outside than inside on an average normal basis. So the ability of a cell to depolarize is critical in neurons and muscle cells, and neurons are in muscle cells, so muscle memory is really neural memory in that. But this electrical signal can give rise to the action potential that we all spoke about and give release to neurotransmitters and contractions and different things like I mentioned to you guys. So at least you get to see some double reinforcement of the information. So voltage-gated sodium channels have two gates: an activating gate that is voltage dependent and an inactive gate that's time dependent. So voltage depends on the charge, and that time gate depends on how long it takes, and then it will close. So the closing will stop the flow of sodium regardless of the activation gate. So that, of course, they have to work together and be in unison. And of course, there's many times where things don't go as supposed to be, and you get diseases and mutations and all these different things. So epilepsy is a common disease that probably everyone has heard about. You probably see when you're watching a movie or playing a video game, it says if you have a history of epilepsy, you shouldn't play it, because all these external stresses when it comes to either, it can be light, it can be hypoxia, oxygen deprivation, it can be mental, emotional, socio-emotional stress, it could be stress of not eating, it could be the stress of being in a fight or flight situation, it can be anything. Fight or flight situation is kind of an umbrella and broad term, because everything comes under that basically, and there's a lot of symptoms that come as a result of that. But epilepsy essentially is a brain dysfunction syndrome caused by excessive synchronization of brain neuron discharge, which basically means that you have just excessive firing of neural impulses going on, and it's not being regulated. And of course, this can go across the corpus callosum, which is the intersection of the two hemispheres of the brain, because the brain is divided into two hemispheres. And usually it's usually two of everything. It's two hippocampuses, it's two amygdalas. So yeah. But in my research, based on the journal I've been looking at And all of the journals you can see are I've mentioned throughout the PowerPoint as well. But the mutation of this specific gene, as you can see, that it's called the SCN1A. I'm not so familiar with this gene specifically, but based on the research, I've came to understand that it encodes for the NAB.
Thank you guys for tapping into the channel of the Nature King. Thank you for finding value in my content, value in my soul, value in my spirit. I have 2 new books that are available in paperback and hardcover on Barnes & Noble, Amazon, and the naturekingbrand.com website. And you can check them out. The link is in the description. Thank you for tapping in to my channel. Don't forget to like, comment, subscribe, share. I have videos coming out nearly every week.